Hi everyone. So let's talk a little bit about DNA and Griffith's experiment as we prep for our team activity that will happen on Friday. So our devotion for today is from James 1:17, very simple. Every good thing given and every perfect gift is from above. So my question for you as we kind of think about this is, do you acknowledge God for your gifts and blessings, or just find yourself blaming God for bad times, or both? So especially in the tough times that are going on, it's important to remember how incredibly blessed we actually are. For every bad thing going on, there's actually a lot of great things going on, and we are blessed people, and God is good all the time. All right, so let's talk about DNA real quick. So this is just a review slide from earlier in the semester, but the product or the properties required of genetic material like DNA is that it has to be able to store complex information. So it has to be stable, and it has to be able to be a code. Right? So within that realm, it has to be able to be replicated. So this is what we're going to talk about in this entire chapter, chapter 9. So how is DNA replicated? So it provides a template for the synthesis of new DNA strands during cell division, right? And this is important for both mitosis, cell division, and meiosis, creating the sex cells like the gametes, the sperm and the egg. We will talk a little bit later in chapter 10 about how this code is used to code for the sequence of proteins. And then finally, genetic material accounts for variation among species. We've talked about this before. So differences in the genetic code, the sequence, accounts for differences in different organisms. But the differences between your cells isn't because of DNA differences. The genome is exactly the same in all your cells. The difference is the proteome, so which genes are being used. All right? So here's our genetic organization in a nutshell. So the nucleotides, the individual nucleotides like A, C, T, and G, that's the smallest part. Codons are groups of 3 of these. So if nucleotides are like letters, codons would be like 3-letter words. And then genes would be like sentences. So a gene is an entire set of codons that codes for a protein. Then chromosomes would be like chapters in the book. So chromosomes have a lot of different genes on them. We have 46 chromosomes total, 23 pairs as humans, and then genome is the entire set of nucleotides and chromosomes. Right? So nucleotides are the basic subunit then. And so we know the 3 components of a nucleotide. We have the phosphate and the sugar, that's the backbone, and then we have the base that forms hydrogen bonds together. And so here are our various bases: adenine, guanine, thymine, cytosine. All right? And so the phosphate and the sugar are the same for all of these. That's the backbone, but it's the base that differs. And remember, we know that A binds to T and G binds to C. So in a double helix, A binds to T, G binds to C. But what's really important to pause and think about here right now is we actually did not know, as scientists in the community as a whole, did not know what DNA did for the longest time. It's only relatively recently, in the last 70 years, that we really figured out that DNA is the genetic material and what this all does. So the amount of progress that's been made in science in the last 70 years since we really started to understand DNA is remarkable. So prior to that, even in 1910, no one really knew what DNA did. And Levine actually had a hypothesis that all the DNA kind of structure was like this, where you had a C bound to a G, bound to a T, bound to an A, kind of in this square format with all those phosphate sugar backbones linked like this. Obviously, this is incorrect, and we know that because there's a lot of experimental evidence. So which of these would be an experimental evidence that shows that Levine's tetranucleotide hypothesis, so this is the tetranucleotide hypothesis, tetra 4 nucleotides linked together hypothesis, which would show that this is incorrect? The concentration of all 4 nucleotides are not exactly 25% each in organisms. Concentrations of A equals T and C equals G. DNA does not have a OH group on carbon number 2 for the pentose sugar. All of the above are evidence. Well, let's think about this. DNA does indeed not have an OH group. That's why it's deoxyribonucleic acid, whereas RNA does, but that really has nothing to do with proving his tetranucleotide hypothesis incorrect. So while this is a true statement, it cannot prove the tetranucleotide hypothesis incorrect. Same here. The concentrations of A equals T and C equals G, we know that to be true because if we have a double-stranded DNA, anywhere you have an A on one strand, you have a T on the other strand. And C binds to G. But that doesn't mean that all four need to be the same. We know that A could be 20% of the genome, which means T has to be 20%, which means the other 60% is divided equally by C and G, so they're 30% each. So even though A has to be relatively equal to T and C relatively equal to G, they don't have to be relatively equal to each other. In other words, C does not have to be relatively equal to T. So A is the answer. Not all four are exactly 25%. If this structure was correct, you would have exactly 25% of every one of these nucleotides all the time. Right? And here's some genomes. Chargraph was the first to really show this. He tested the nucleotide concentrations or amounts in various genomes: E. coli, yeast, sea urchin, rat, human. All of these have double-stranded DNA genomes. And you can see that A, T, C, and G are not always 25%. All right, let's look at yeast here. In fact, you might notice that A and T are not exactly the same and G and C are not exactly the same, but that actually is because the various ends of the chromosomes can be a little bit different. But in reality, what he noticed is that the ratio of A to T is almost always near 1, and the ratio of G to C is almost always near 1. And that's because A is going to be equal to T, and G is going to be equal to C in all these genomes. All right, so this is where we're going to start this Friday. People had no idea what the genetic material was. They knew about genetics. They knew that you could inherit traits. All right, so the idea of genetics had been around long before we actually understood what DNA did. And in fact, up until probably the early 1930s, 40s, and 50s, people really thought that protein was going to be the genetic material. Well, why? Well, all of these are reasons. DNA is pretty simple. There's only 4 nucleotides, right? Whereas proteins are very complex. Even the structure of DNA, that double helix, is very simple, whereas proteins we know can fold in an almost infinite number of ways. So proteins are more complex. We are complex organisms. Everyone figured that the genetic material must be very complex. DNA was just not complex enough. It must be proteins. Part of that complexity comes from the fact that proteins have 20 amino acids, 20 building blocks. DNA only has 4. So surely to code for especially the humans, the amazing human race, you would need complexity that requires 20 building blocks. 4 would just not be enough. People already knew that proteins were the workhorses of the cell. They are the enzymes, they are the membrane proteins, all kinds of things. So surely they could carry information as well. So these are all reasons that people really thought that protein was inevitably going to be proven to be the genetic material. Obviously, we know that's not the case. It ended up being DNA, but this was the major argument at the time. So that was the problem. How can DNA be the genetic material if it's very simple, right? Just 4 nucleotides, and it has a very regular structure, that double helix. All right, so let's get into some of the experiments that really started to allow us to understand that DNA was the genetic material. The first one, and the one you really have to understand, is transformation. So here we have Griffith in 1928, and he used Streptococcus pneumoniae. So this is the bacteria that causes pneumonia. But there's multiple types of pneumonia, and 2 important ones here is smooth pneumonia and rough pneumonia. So smooth pneumonia, this bacteria has genes that allow them to secrete a protective polysaccharide coat. So that sugar coat on the outside makes them appear smooth in culture, versus the rough does not have that protective polysaccharide secretion, and they kind of look more rough. This is also very important in illness, so pathology. Because of the protective polysaccharide coat, smooth pneumonia is able to evade our immune system much better, and therefore our immune system has a harder time getting rid of smooth pneumonia, and smooth pneumonia can be very lethal. Right? However, rough pneumonia doesn't have that protective polysaccharide coat. Because it doesn't have the genes to make that polysaccharide coat, our immune system is able to recognize it and attack it pretty quickly, and so rough pneumonia really doesn't make you very sick. It can be cleared by the immune system. Right? And importantly, the smooth or rough traits are passed on to progeny. So this is genetic, right? If you're a smooth bacteria, your progeny, your daughter cells, are going to also have the genetic material to make that polysaccharide coat. They're going to be smooth as well. If you're rough, you don't have that gene. You can't pass on that gene to your progeny, and your daughter cells are going to look rough as well. All right, so here was Griffith's experiment. So he injected mice with smooth bacteria, and we know the smooth bacteria is the virulent one. That's the one the immune system can't get rid of. And so as expected, this caused the mice to die. Right? This is the pathogenic strain. When he injected rough bacteria, the mouse lived because this is not pathogenic. The mouse's immune system was able to get rid of it. All right. When he killed the smooth bacteria with heat, well, as you might expect, now the heat-killed bacteria, when injected into mouse, the mouse lived, because basically you've just injected non-live bacteria. And the bacteria not alive can't divide, can't do a lot of things, the mouse is able to live. But here was the surprising part. So he then took rough bacteria, so live rough bacteria, but heat-killed smooth bacteria. So let's think about this. We know that rough bacteria injected doesn't kill the mouse. We know that when you kill the smooth bacteria and inject that, it doesn't kill the mouse. However, when he injected the rough bacteria that was incubated with the heat-killed smooth bacteria, the mouse did indeed die. And when they cultured the bacteria from the mouse, they found that the bacteria looked smooth. So even though all the smooth bacteria was killed, it could not have lived, it could not have divided, this mouse had no smooth bacteria in it when you injected heat-killed smooth bacteria. This bacteria killed the mouse and looked smooth. So what happened was this process called transformation. And this is the important part. The rough bacteria absorbed the genetic material from the dead S strain. So in other words, the genetic material from this dead smooth strain was released into the environment, and the rough strain picked up that genetic material, and then because of that started having the gene to make the polysaccharide coat. So basically the rough bacteria was transformed into smooth. So the ability to pick up that genetic material from the environment is called transformation. In other words, the rough bacteria picked up the genes from the dead smooth bacteria, and now that they had those genes, they could make the polysaccharide coat and kill the mouse. Griffith himself, however, never did discover what that chemical was. So there's a chemical being released by this dead smooth bacteria that's picked up by the rough. And that's the genetic material. But Griffith didn't figure out which it was. It was narrowed down to our 4 macromolecules. It was narrowed down to being protein, as most people would have predicted, lipids, carbohydrates, or DNA or RNA. So what we are going to do in lab, or in class, is determine the transforming biomolecule. We're going to look at the evidence that determined that DNA was the transforming biomolecule. But what I want you to do is think about how you would do this. Right? So you now have this experiment where you can incubate dead smooth bacteria, or genetic material released by the smooth bacteria, and you can incubate it with rough bacteria, and the rough bacteria, if it picks up the genetic material, will start to look smooth. And we have 4 candidates. How would you determine which of those 4 candidates was the one that was allowing the rough bacteria to pick up the genetic material? Think about it, and that's where we're going to begin on Friday.